The first point you have to remember is the name of MHC molecule. The full form of MHC molecule is Major Histocompatibility Complex. During the early studies, those scientists who are working on tissue transplantation, they are searching for a molecule. What happens when a donor donates tissue or organ to a recipient? There is a chance of the acceptance or rejection of the tissue or organ. So acceptance or rejection, there are two options. So scientists are working on a molecule which is responsible for these acceptance and rejections, and they found a molecule called Major. Major means this molecule has a major role in histo means tissue, compatibility means whether the recipient will accept or not or reject the tissue. Compatibility means whether the tissue is compatible for the recipient. So they found a molecule that has a major role in the tissue compatibility, so they name it Major Histocompatibility Complex. Later it was known that MHC is responsible for presentation of antigen. So actually MHC represent antigen. Okay, antigen can be of different type like carbohydrate, lipid, protein in nature. MHC molecule only represent antigen that is peptide in nature. And another important thing is a antigen consists of many epitope or antigenic determinant. I am showing you. Let's assume this is an antigen. A antigen actually not responsible for the binding of antibody or T cell receptor. There are some portion of the antibody antigen like these are called the epitope. So this is called epitope or antigenic determinant. So in an antigen there may be different type of and many number of epitopes. Antibody bind with this molecule called epitope or antigenic determinant, and this antigenic determinant can be of two types. One is called conformational, which an epitope are in conformation like alpha helix or beta sheet are called conformational epitope or conformational antigenic determinant. And some epitopes present in linear conformation or denatured conditions. When the epitope is denatured, it is called linear. MHC molecule represent antigen that is peptide in nature, also that is linear in antigenic determinant. Linear. The next point is there are three type of MHC molecule called MHC I, MHC II and MHC III. This MHC I and MHC II actually represent antigen, but the MHC III gene actually produce some proteins that are the component of complement protein like C2, C4, factor B, and some molecules that actually induce inflammation like TNF alpha and lymphotoxin alpha, or it is also known as TNF beta. The next point is this class 1 MHC molecule actually represented by all nucleated cells of our body. All nucleated cells, those cells have nucleus, can represent. RBC do not have nucleus, so they do not express MHC I molecule. I want to give you some example of cells that express very low or undetectable level of MHC I molecules like liver. So generally liver transplantation do not fail. A recipient when receive or actually receive organ like liver, it do not actually reject because liver cell express very low amount or undetectable level of MHC I molecule. And in the beginning I want to mention MHC molecule is actually recognized by only T cell receptor. T cell receptor bind with MHC and peptide complex. Okay, so recognize MHC and antigen that is peptide in nature complex, both these molecule together, and this MHC1 is recognized by particular cell called TC cells. The T cell can be broadly categorized into two population called TC cells and T helper cells. TC cells means cytotoxic T cells. These cytotoxic T cells express a specific co-receptor called CD CD8 positive. That means this cell has CD8 co-receptor. They are designated as CD8 positive T cells. Cytotoxic T cells perform its function by killing directly the cell. It is actually kill a virally infected or tumor cell. But helper cells do not kill directly. It actually helps other cells to kill the infected cell. So the function of these two cells is different, and they actually express two different co-receptor. Like TC cells express CD8 co-receptor and T helper cell express CD4 co-receptor. So they are called plus. This means they have this co-receptor molecule. Then MHC II molecule is expressed by very few cells called professional APC, means antigen presenting cells. These cells are professional in presenting of antigen. Example of these cells are macrophage, dendritic cell, B cell. But some cells like endothelial cell and thymic epithelial cells can express MHC II molecule. The MHC II molecule is recognized only by T helper cell that express CD4 positive cells. TC cells or cytotoxic T cells cannot recognize MHC II, and T helper cells cannot recognize MHC I. They are restricted to one type of MHC molecule. The next point is the MHC molecule or gene is the most polymorphic gene of mammalian genome. So MHC molecule is diverse in nature. So MHC is diverse. The reason behind this diversity is one, polymorphism. Polymorphism means, let's assume this MHC molecules is expressing from a gene called A. Polymorphism means alternative allelic form. So this A gene can be represented by A1 gene, A2 gene, A3 gene, and so on. That means this A gene can be of different allelic form. They all perform the same function but have a difference in the structure. So this is called polymorphism, when a gene present in different allelic form. Now for the MHC diversity, this is not only the reason. There is another reason that gives the diversity to the MHC called polygeny. Let's assume this A gene is producing different polymorphic allelic form. Also this MHC molecule can be encoded by another gene called B, another gene called C, D, and so on. And all these gene like B cells also contain the polymorphic or alternative allelic form like B1, B2, B3. Now you can imagine how much diversity can be produced from these two mechanisms. One is polymorphism, alternative gene or allelic form, and polygeny means different many alleles are actually expressing the same molecule MHC. Another reason is present, that is during my another lecture I have already mentioned that antibody and T cell receptor go through a process called allelic exclusions. What is allelic exclusion? I want to mention here. When a B cell express antibody from its mother chromosome, it will inhibit its father chromosome. So if this is a B cell, and if this B cell is producing its antibody from its father chromosome, this particular B cell will not produce antibody from its mother chromosome. It excludes its mother chromosome to produce the same antibody. This is called allelic exclusion. This is also true for T cells. If a T cell is producing its T cell receptor from its mother chromosome, it will exclude the father chromosome to produce the T cell receptor. This is called allelic exclusion. But MHC do not go through allelic exclusion. Allelic exclusion, they actually inherited codominantly. That means in a single cell, let's assume this is a cell, in a single cell both the father and the mother chromosome can produce MHC molecules. That means codominant. Number 7 is this polymorphic gene and the polygeny. For the MHC molecule, these all genes are present very close to each other. So when they inherited from the father chromosome or mother from sperm or ovum, they actually inherited as a set. Like A gene is coming from another chromosome or another location, B gene coming from another location. No, they come as a set. This is called haplotype. So this means set of MHC gene. Okay, another important point I want to share with you, that in case of antibody can recognize only particular antigen, or T cell receptor can actually recognize a particular antigen. But MHC molecule, one MHC molecule can bind many peptide, and also one peptide can bind many MHC molecules. So MHC molecules is promiscuous. It is not very specific. So it can bind with many different peptides, or one peptide can bind with many different MHC molecules. I will discuss about this in more details when I will explain MHC I and MHC II very specifically. Today's class is on the basic concepts of MHC molecules. Okay, the last point, the major function of MHC molecules, another major function called self-MHC restriction for two cells. One is T helper cell and for the TC cells.